spreading depolarization triggers pro- and anti-inflammatory signaling, a potential link to headache. Cortical spreading depolarization, CSD, the neurophysiological event believed to underlie aura, might trigger migraine headaches through inflammatory signaling that originates in neurons and spreads to the meninges via astrocytes. This study aims to elucidate the progression of post-CSD inflammatory signaling and its resolution in neurons, astrocytes, and microglia in mouse brains. CSD was triggered optogenetically or by pinprick, high mobility group box 1 release, caspase-1 activation, and cell-specific activation of nuclear factor kappa B pairs, along with ensuing transcriptomic changes, were evaluated using immunofluorescence, Western blotting, co-immunoprecipitation, fluorescence resonance energy transfer analysis, and cell-specific transcriptomics. Immediately following CSD, neurons release high mobility group box 1, HMGB1, in extracellular vesicles. This is accompanied by caspase-1 activation, which peaked 1 hour post-CSD and diminished within 3 to 5 hours. This suggests that pro-inflammatory stimuli driving inflammatory signaling decreased within hours after CSD. Pro-inflammatory nuclear factor kappa B P65-P50 pairs, along with anti-inflammatory C-rel P65 pairs, were detected in astrocyte nuclei shortly after CSD. However, 24 hours post-CSD, the former had disappeared, whereas the latter persisted, indicating a shift from pro- to anti-inflammatory activity in astrocytes. Pathway analysis of cell-specific transcriptomic data confirmed pro-inflammatory transcription in astrocytes and microglia, 1 hour post-CSD, whereas no such activity was observed in neurons. Detailed transcriptomic analysis with Bayesian cell proportion reconstruction revealed that microglia exhibited transcriptional changes trending towards an anti-inflammatory profile, along with upregulation of several chemokines and cytokines, such as tumor necrosis factor. Our findings suggest that microglia might play a role in supporting the inflammatory responses in astrocytes through the release of these mediators. The upregulation of genes involved in chemotaxis, such as CCL3, and spine pruning, such as C1q in microglia, implies that microglia might contribute to synaptic repair, while inflammatory signaling in astrocytes could potentially modulate meningeal nociceptor activity through an extensive astrocyte end-feet syncytium, abutting subarachnoid and perivascular spaces, although direct evidence remains incomplete. This nuanced understanding of the inflammatory response in CNS cell types highlights the intricate cellular interactions and responses to CSD. Following a single CSD, distinct transcriptomic responses occur in neurons, astrocytes, and microglia, driving inflammatory and anti-inflammatory responses, potentially contributing to headache initiation and resolution.